Hi guys, we're back with a proteins video. So this is all about proteins. It's chapter 2.4 in the book. So first we're going to start out with the types of proteins that we have. So they're made up of amino acids. That's the monomer. So if you break down a protein to the smallest element, the smallest component, it'll be an amino acid. So first we're going to talk about hydrolysis and condensation. So if you watch the two other videos before this one, about 2.3, when it talks about carbohydrates and lipids, we talked about condensation and hydrolysis in terms of those two macromolecules. But now we have to apply the same concept to proteins. So let's draw out our protein. We have our amino group over here, N. We have two C's, an H, the R group, put that in red. And over here we have our carboxyl group. And over here we're going to have another one of these. We're going to have H, H, to an N, C, C, H, R, and carboxyl. Okay, so what's going to happen here is we always have to release one water. So the ratio here is actually perfect. We have two H's and an O, which will give us H2O. So if we want to redraw this into the final dipeptide, is what it's called, it will look something like this. So we're going to get rid of these, because these will turn into a water molecule. These two will connect. Let me put that closer. These two are going to connect, and unlike the other two macromolecules, they actually will not have an oxygen in the middle. And this oxygen over here will remain, and the hydrogen here will remain. So they're not going to have anything in the middle. It's just the C is going to connect to the N. That's it. And this bond is going to be called a peptide bond. So as we saw in the other two videos, two glucoses combined will be a glycosidic bond. Two lipids combined form an ester bond. And now we're moving on to proteins, which form a peptide bond. So these two would be called a dipeptide. When you add more, it'll become a polypeptide. And it'll look like a chain. A chain of peptides will be a polypeptide. Okay, so now that we got those terms out of the way, we have to start looking at the stages. So stages of protein. So is a polypeptide the same thing as a protein? No. The answer to that question is no. Polypeptide is not a protein. Why is that? Because a polypeptide is just a chain of amino acids, whereas a protein has a structure and has a function. So the first stage we're going to talk about is primary structure. So a polypeptide in primary structure is going to look something like this. Pretend those are amino acids. It's not really going to have much shape. It's just a polypeptide with peptide bonds between them, and it has no function. However, then we start moving, so this isn't really classified as a protein. Then we move into secondary structure. This is when we can finally classify some of these polypeptides as proteins, because once they establish this shape, that's it. There's no more development for them. They're stuck in this shape. Some examples of this are keratin and collagen and yeah. So secondary structure, it's composed of this shape, like a squiggle, and it's composed of this shape. So we call this a beta sheet. And we call this an alpha helix. These are just the names. So when you see the squiggles like this, beta sheet, squiggles like this, alpha helix. So if you want to describe a polypeptide in primary structure, you call it a chain of amino acids, somewhat linear. And secondary structure would be beta sheet and alpha helix. So these two can be classified as fibrous proteins. Okay, I'm going to write that here on the side. Fibrous, yeah. So this is the fibrous type. It's long and narrow, and we're going to go into some characteristics later on. Okay, so fibrous. Now, moving on to the stage number 3. It's called the tertiary stage. Tertiary structure. Now, a tertiary structure is when proteins kind of establish like what they're doing in life, you know? It's like these are children. There's a baby, like a toddler. Tertiary structure, you kind of know what's going on. And, well, it looks kind of, you know, foldy. Not exactly like this, but it has the folds. So an important term to use here, it's folded. So when it comes to proteins, there's something you should keep in mind, that when it's folded, when it has a shape, it has a function. So over here, is there shape? No shape. Here, they're starting to become a shape, which over here I'm going to write keratin and collagen are found in secondary structure. Tertiary structure is when they finally start to have shape, so they have bigger function. They have more stuff to do because they have a shape. So that's tertiary structure. And last, we move on to our quaternary structure. So what is a quaternary structure? Well, in simple terms, it is a bunch of tertiary structures linked together. So over here I have 1, 2, 3, 4 tertiary structure proteins combined to form a quaternary structure. Okay, so now we're going to classify these two into a category that will simply be called the globular type proteins. So these are classified as proteins. Secondary structure can be classified as a protein if that's where the development ends, but these are the real proteins we're talking about. Now, why have I mentioned the fibrous and globular proteins? Fibrous, globular. So fibrous have a structure. Let's write structure. It's long and narrow, as we can see over here with the polypeptide. Oh, let me erase these. With the polypeptide and these, and the secondary structure. And the globular has a spherical shape, as you can see, and it's kind of globular, like it's a circle kind of when it's folded around. So what is folding due to? It's due to interactions between the amino acids and how they bond and how they connect. So, okay, we got the structure down. Now, we have functions for these. So the functions could depend on what the protein is. Usually fibrous proteins are for more aesthetic purposes. So we have here keratin. We have collagen. And over here, in the globular, we have stuff like hemoglobin, transports oxygen. We have something like an enzyme. You have amylase. We have hydrolase, which we took. Hydrolase, okay. Yes. So, yeah. Those are some examples. Now, moving on to... let's move on to heat and some things that can denature. So I'm sure you've heard this term in class. It's a very important term that you must know. So denaturing a protein so I said for a protein to be functional, so a happy protein, we need a shape so it can have a function, right? If there's no shape, there's no function. So when we denature a protein, there will be no shape. Therefore, there will be no function, or less function, or a not as well function, okay? That's the gist. So how do you really denature a protein? Well, there are three main factors. Whoa, 3. That's great. 1, 2, 3 main factors to denaturing. How to denature. This is like a how-to guide on how to denature a protein. Okay, first we have excessive heat. So if this protein isn't used to having all this heat or cold or temperature, it will damage its function. It will damage its shape, it'll damage its function, it'll make the protein absolutely useless in your body, and you can't go back on the biological factors that have to do with it. Next, we have pH levels. So pH is basically acid. You're checking the acid levels. For example, in your stomach, the enzymes that live in your stomach have to deal with a lot of stomach acid for you to be able to break down your food. So those proteins are less sensitive to pH levels than, for example, the enzymes that are in your blood or other enzymes. So it doesn't affect all of them. This is very subjective. Well, enzyme-jective. Okay, last we have lead and copper. These will affect. I'm not quite sure how, but they do affect. And yeah, basically, that is it for proteins. But there's one last thing I would like to tell you, which are two terms. The first term is a proteome. Proteome. And the other term is a genome. So we all know that, well, I'm going to tell you that to produce a protein, let's say this is your cell, and this is your nucleus, and this is going to be our new friend, the ribosome. Label this ribosome. This is the nucleus. So DNA is stored and produced in the nucleus, right? And it's basically the blueprint of your whole body. So everything, all your traits and genes, everything is found in your nucleus. So what happens is the DNA can't leave the cell. It's grounded, basically. It can't leave the cell. So it takes messenger RNA and it leaves the cell and it goes to the ribosome, where they actually produce the proteins in a process called translation and transcription. It produces the proteins which then are able to work in your body. So where are the proteins produced? In the ribosome. Okay, so now speaking of that, the genome is the total, is all the genes in your body, in our body. It's a collection of genes and traits and characteristics. Everything is in the genome. So those genes, to make a trait, they have to code for a protein. The protein then will actually form the trait in your body. So a genome will lead to the proteome, which is all the proteins produced by our body. So basically, the genome codes for the proteome, and obviously the proteome will be larger than the genome because for every one gene you produce like a lot of proteins. So let's say you have like apples on the trees, cars in the garden. A, C, C, G. This is like the codes of the nucleotides. A, T. Okay, let's say this codes for one protein. You can form all these proteins just from this simple code in the genome. That's how you always know the proteome will be larger than the genome. That's it for proteins. Thanks for following along. Let me know if you need any help and comment if you need help. Thank you and farewell.